Hello my dear students, this is Dr. 𝗲𝗵𝗵𝗺, your biochemistry mentor. In this video, we are going to discuss about the metabolism of chylomicrons. Now before starting up with the video discussion, we should know what are these chylomicrons. Right. So basically, chylomicrons are the type of lipoproteins. Right. What are lipoproteins? It is clear from the word itself, lipid plus protein. Right. And if we talk about the structure of a lipoprotein, it has an outer protein covering and inside it has lipid and protein. So basically, polar part is projected towards outside. Polar is outside and non-polar is inside. Okay. So what is the requirement? Because if we talk about lipids are non-polar molecules which are not soluble in water. So whenever they have to be transported, they always seek a carrier protein. So the protein part is known as a apoprotein, and this apoprotein, when it binds with a lipid, it becomes a lipoprotein. Now coming over to the chylomicrons, these chylomicrons are the largest among the lipoproteins. Why? Because if we talk about the size of chylomicrons, they are having this type of size, biggest. Now why they are having biggest size? Because they have maximum TGs, maximum triglycerides. If it has maximum fat content, so automatically least will be the protein content. As simple as that. Right? Because they are laden with fats, so small amount of protein will be required just to push them. And what happens is, whenever we talk about the metabolism, we know that chylomicrons, whatever things they are having, it will be, it will start from the intestine, they have to undergo absorption. So always remember, lipoprotein metabolism revolves around three organs: intestines, adipose tissue and the liver. Because liver being the kitchen of the body, adipose tissue, adipocytes, they are linked with obviously the fats, and intestine from where the circulation or absorption starts. Okay. So these chylomicrons have actually, if we talk about the apoproteins associated with this, they have Apo B-48. Very, very important. This Apo B-48 is a specific apoprotein for chylomicrons. And Apo B-48 is synthesized in the body by a process of RNA editing or processing. Right. So Apo B-48 is the specific apoprotein for chylomicrons. Second is Apo C-II. Now this Apo C-II is the activator of, or it activates the enzyme lipoprotein lipase. And third is Apo E. And Apo E is responsible for receptor-mediated endocytosis. Receptor-mediated endocytosis. We are going to discuss what are the functions of these apoproteins, how they are going to transport these TGs, and how we are extracting the fatty acids and glycerol from these TGs. Because ultimately what we have to get is, we have to break these TGs into fatty acids and glycerol. and these fatty acids will then undergo oxidation and glycerol will also be utilized. That is what we have studied, कि glycerol will undergo the formation of glucose. Okay. So now what happens here is, first of all, the specific apoprotein is Apo B-48, which is produced in the body by a process known as RNA editing. Apo C-II, which will activate lipoprotein lipase. So just see what is the nature of this enzyme. Commonly we will use it as LPL. So I will be using this terminology, LPL. And Apo E is receptor mediated endocytosis. Means what will happen is, whenever we have to extract the amount of triglycerides, whatever is there, for that liver needs a kind of a recognition. So liver is going to accept Apo E, which is coming with these TGs, right? So first of all, these chylomicrons, if we talk about the metabolism, they help in the transport of exogenous TGs. This is very, very important. Exogenous, or we can call them as dietary TGs. Right. And when the chylomicrons leaves the intestine, they are known as nascent chylomicrons. So I can say that nascent chylomicrons will be recognized by what? They will be recognized by Apo B-48. Apo B-48 is already there. So if I say this is the intestines, right? And if I say this is the adipose tissue, and say this is the liver, because it will be between these three. Now see what will happen the moment chylomicrons are leaving this. This is a chylomicron and there are three apoproteins associated with this. One is Apo B-48, but because I told you it is the specific apoprotein for this. Apo C-II will come and Apo E will come. And this chylomicrons will be full of triglycerides. So TGs will be present here. Right. Now the question is that this will acquire more TGs like this, and it will become a mature chylomicron taking these three apoproteins like this. So this becomes mature chylomicron. I would write like this, mature chylomicron. Right. Now see, this is Apo C-II and this is Apo E. Now I told you Apo C-II का function क्या है? It is an activator of LPL. Now what is this LPL? LPL will be here like this in the endothelial lining. So this LPL is going to accept these mature chylomicrons. The moment this enters here, whatever TGs are there, these TGs will be broken, these TGs will split up. Means the function of LPL is to extract the TGs from chylomicrons and VLDL to send them to the adipose tissue. I'll just repeat it again. Nascent chylomicrons are the ones which are produced the moment they are leaving the intestine. Apo B-48 will be specific and it will bind with this. Apo C-II and Apo E will, it's like they will come in the way. So it is going to take all the TGs together, whatever is there, and it will be bigger in size. It will be known as mature chylomicrons. Now LPL is already lining the endothelial cells. So what will happen is LPL is going to extract these TGs from the chylomicrons. So what happens here is these TGs will be taken up. So I can say that these TGs will split up into fatty acids and glycerol. And what is left here is, the left part is the remnant chylomicrons. Right? And if we talk about this, we can say that this fatty acid will combine with glycerol here, and it will be forming TGs, and it will be saved in the adipose tissue. Okay. Now, if we talk about glycerol, this glycerol will enter in the liver, where it will be metabolized. Now, why glycerol will enter in the liver? Because glycerokinase is absent in the adipose tissue. In the adipose tissue. Right. Now, these remnant chylomicrons, now again Apo B-48 will stick to it. Apo B-48 is not going to leave this, and Apo E is there. Okay. Now, the last step would be, here antenna shaped molecules will be there like this. These antenna shaped molecules are going to be recognized by this Apo E receptor. That is why I told you it is a receptor mediated endocytosis. Okay. So what happens here is receptor mediated endocytosis. And this remnant chylomicron will be accepted here, and where it will be completely degraded to form again fatty acids and glycerol. Right. So this is what is the metabolism of chylomicrons. First is, I can simply say that they help in the transportation of exogenous or dietary TGs. Second step is, nascent chylomicrons will be recognized by Apo B-48, then Apo C-II and Apo E will come from HDL. Okay, so they will form a team, and they are going to form the mature chylomicrons. Right. Then next step is, simply we can say the fifth step would be LPL is activated because Apo C-II is there. So Apo C-II is going to activate LPL. This is very important. The moment LPL is activated, LPL will take the TGs from the chylomicrons. They will just extract. And that is what is the difference between this LPL and HSL. Both enzymes are different. Lipoprotein lipase is a different enzyme, and hormone sensitive lipase is a different enzyme. So I can say LPL is actually a kind of, anabolic enzyme. But yes, it will just take the TGs out from the chylomicrons. That's it. Okay, then what will happen? The leftover will be remnant chylomicrons. Right. Now, glycerol will simply enter into the liver for again It will undergo the formation of glucose, or you can say it can enter glycolysis. And this remnant chylomicrons are finally accepted by the liver to undergo receptor mediated endocytosis. Which is very, very important, right? So, if we talk about LPL, we should know that what is the function of this LPL and where it is present. So, there are two enzymes basically. One is LPL, second is HSL. This stands for lipoprotein lipase. And this stands for hormone sensitive lipase. Right. LPL and HSL. Now, what is the basic difference between the activity of these two enzymes? First is it is present in the endothelial vessels. HSL is present inside the adipose tissue. Right. Second is lipoprotein lipase is an anabolic enzyme. This is very important. And HSL is a catabolic enzyme. And third is lipoprotein lipase is always activated by insulin. This is very, very important. It is always activated by insulin. So, obviously, if it is activated by insulin, it will be inhibited by glucagon, because glucagon and insulin, they work antagonistically. And if we talk about this, HSL is always activated by glucagon, by epinephrine, and third is thyroid hormones. And the last difference is that lipoprotein lipase takes TGs from chylomicrons and VLDL. And this basically degrades TGs, takes TGs into fatty acids and glycerol. But they never react on the second form of TGs. Okay. So, always remember, if we talk about lipoprotein lipase, it will be always activated by C2. And if we talk about hormone sensitive lipase, it will be always activated by glucagon or epinephrine. So, it is altogether different. So, whenever we talk about the metabolism of chylomicrons, or whenever we talk about the metabolism of all the lipoproteins, the basic enzyme which is going to act on these lipoproteins would be lipoprotein lipase. Because always remember, if we talk about, जैसे for example, if I talk about hormone sensitive lipase, right, hormone sensitive lipase, it will do what? It will break these triglycerides into fatty acids and glycerol. Okay, these fatty acids will then undergo oxidation, whether it is beta oxidation, whether it is alpha oxidation, or you can say omega oxidation. So HSL will always degrade whatever triglycerides because they are the esters of fatty acid and glycerol. So it is going to take this, and glycerol will automatically, since I told you glycerokinase is absent in the adipose tissue, so it will undergo formation of 3-phosphoglycerolaldehyde like that, or it can form dihydroxyacetone phosphate. Right? That is why we always say कि hormone sensitive lipase is inhibited by insulin but activated by glucagon, and that is why we say it is a catabolic enzyme. And we know that insulin is an anabolic hormone. On the contrary, if we talk about the lipoprotein lipase, it is forming that. It is simply taking TGs out from that. It is not degrading the TGs. It is simply taking the lipoprotein from the vicinity of chylomicrons and VLDL, because chylomicrons are responsible for the exogenous pathway and this VLDL is responsible for the endogenous pathway. And this is the basic difference between exogenous triglycerides and endogenous triglycerides. Okay, so this is the metabolism of chylomicrons, or what you have to remember here is the most important point which you have to remember here is Apo B-48 is the specific apoprotein for chylomicrons, which is produced in the body with the help of RNA editing or processing. Apo C-II is again used as a apoprotein, which is an activator of LPL enzyme, and Apo E is responsible for receptor mediated endocytosis. And what happens is they are required for the transportation of exogenous TGs. And the moment they leave the intestine, they are known as nascent chylomicrons and they are picked by Apo B-48. Once they are picked by Apo B-48, Apo C-II and Apo E will also appear, and they are going to add more and more of this TGs to form mature chylomicrons. And these mature chylomicrons, as you can see that lipoprotein lipase is there, which will extract these TGs out of this. And it will be converted into remnant chylomicrons. Remnant means whatever is left over. And these remnant chylomicrons will be accepted by the antenna-shaped molecules and responsible for receptor-mediated endocytosis. That is what I have written. Apo C-II will activate. LPL will just take the TGs from the chylomicrons, and what we are left with? We are left with the remnant chylomicrons, and finally they are accepted by the liver to undergo this. So this is how the exogenous TGs are accepted by the liver, right? So this is all about the metabolism of chylomicrons, and always remember one thing, that if we have to differentiate chylomicrons from HDL, the basic difference is that chylomicrons have maximum TGs and least protein content. And if we have to separate them by electrophoresis and ultracentrifugation, because least protein, so obviously they will be least dense. Okay. And the specific apoprotein is Apo B-48. This we have to remember, because after finishing up with the metabolism, we are going to study the different type of hyperlipoproteinemias, which are very, very important to understand. To have the understanding of different type of hyper and hypolipoproteinemias, we should be familiar with the metabolism of chylomicrons. So this is all about the metabolism of chylomicrons. Thank you so much.